I am going to introduce our first speaker this evening is Bill Hack, and he is the founder and CEO of Cadex Genomics, excuse me.
Greetings. Cadex Genomics is a molecular diagnostics company that has developed a blood-based tumor burden platform which runs on PCR and has multiple applications in the field of oncology. Hi, my name is Bill Hack. I am the CEO and one of the co-founders of the company. The problem we address with our first product is to improve on the current standard of care for assessing the effectiveness of therapies delivered to cancer patients with stage 4 solid tumors. Currently, imaging is used to assess whether a patient's therapy is working. Imaging is typically performed in 8 to 12-week intervals. The problem with this approach is that when the therapy is not working, the patient's tumor is left to grow unchecked for weeks. Additionally, the patient is receiving all the toxic side effects of those ineffective treatments and none of the intended benefits. And finally, it is a tremendous waste of healthcare resources to deliver multiple cycles of ineffective treatment. Many of these treatments cost tens of thousands of dollars, and patients pay an increasingly higher portion of those costs. Our solution to this problem is Alebrix. Alebrix is a real-time, blood-based therapy monitoring tool. We compress the time it takes to evaluate the effectiveness of a patient's treatment from 12 weeks to just 12 days. And most importantly, after just a single cycle of treatment. Alebrix has the potential to improve patient health outcomes while significantly reducing the cost of cancer care. This is a $3.5 billion market opportunity. The way our technology works is conceptually very simple. When cancer cells die, their DNA is shed into the blood. This is called cfDNA or cell-free DNA. All other things constant, the larger a tumor, the higher the concentration level of cfDNA in the blood. So by comparing the cfDNA concentration level in the patient's blood prior to treatment with the cfDNA level 12 days after treatment, we can determine if a patient's tumor is growing. The reason our test is so accurate and precise is we target a section of the DNA called retrotransposable elements. This part of the DNA has 1,800 copies in every cell, giving the assay unmatched analytic sensitivity. To make the assay cancer specific, we filter out non-cancer cfDNA by exploiting the fact that cancer cell fragments, DNA fragments, are shorter than non-cancer cfDNA fragments. Our approach has many advantages over approaches of other cfDNA companies. These are, we require no changes in the patient workflow. Our technology is extremely cost effective. The cost of goods for analyzing both blood draws is under $50. And we are the only company with data demonstrating we can monitor therapy effectiveness in real time. We have studied our technology in over 175 patients across 3 studies. The first of these studies is a proof of concept study comparing healthy patients against treated and untreated stage 4 colorectal cancer patients. The results demonstrate that our technology can distinguish between these 3 cohorts with an area under the curve of 0.989. The second study, which we conducted with MD Anderson, determined that within 2 to 3 weeks, Alebrix can identify 63% of patients with progressive disease at 8 to 12 weeks using imaging. Most important, there were no false positives, 100% specificity. And in our most recent study, we were able to identify 80% of patients who were classified as having clinically relevant progressive disease using imaging at 8 to 12 weeks. Again, with no false positives, 100% specificity. Here are the key company milestones. Looking forward, by the end of this year, a Librix will be finalized and will be ported to a point-of-care PCR platform that will give us tremendous scalability and the ability to easily move to markets outside the US. The developer of this point-of-care platform is a strategic partner for us who is funding large portions of our R&D. They have already expressed an interest in acquiring the company. By the end of this year, our first clinical validation study will be completely enrolled. The data from this study will be evaluated by the end of Q1 of next year, at which point we will commercially launch a Librix for stage 4 colorectal cancer. By the end of next year, we will expand the Librix into lung and breast cancer. We will also receive Medicare coverage by the end of next year, which we believe is the optimum point to exit. Here's the team you will be investing in. All 6 of us has 20 plus years experience in life sciences, bringing novel drugs and diagnostics to market for cancer patients. We have worked at companies like Genentech, Veracyte, Invitae, Castle Biosciences, and others. This experience is in fields such as R&D, clinical operations, medical, sales, marketing, and reimbursement. I thank you for watching, and I look forward to hearing from you soon. Thank you.
Bill, thank you so much for your presentation tonight. You know, this seems like a really, really exciting technology. And as you mentioned, the idea that patients really would know a lot faster whether their treatment is working, patients and providers, I should say, as well as the fact that they, you know, hopefully would suffer less side effects. We all know that cancer treatment is not a fun process to go through. So very exciting. I'm going to start off with a couple questions, and then I know those in our audience are putting some questions into our Q&A function. Again, that is just to the right of the chat. I would love to take your questions here in just a minute, but I'm going to start off with a few of my own. So you mentioned that colorectal cancer, breast cancer, and lung cancers are kind of first in your kind of pathway. Can the technology be used for all types of cancer?
We believe so. The you know, it's generic. The marker that we're looking at is fairly generic to cancer, meaning we're looking at these retrotransposable elements, non-coding regions of the DNA. And so those are shed by every tumor type. And so we think the assay will work across all tumor types. But the proof is in the pudding, and we need to do further studies to prove that that is the case. So for now, we do have evidence for lung, breast, and colon. And we'll need to do additional studies to actually demonstrate that it works across all tumor types.
Wonderful. And you talked a little bit about the technology itself. And my understanding is you're looking at kind of the length of the cfDNA markers to determine if this is tumor or non-tumor cfDNA. Can you talk a little bit more about that?
Of course, and that would be a great question, by the way, to re-ask when we break into the group the table
networking.
Yeah, the networking. Our chief scientist will be there, and he can answer this with far better precision than I can. But that is correct. So there's really 3 things that that really ensure that we're looking at cfDNA from tumor types, from the tumor as opposed to non-cancer cells. And the first thing we do is we can sort of cancel out the
the background noises, if you will, because we do take a blood draw before therapy. And if there is background noise from non-cancer DNA, and those cancel out because we compare with, with a blood draw after therapy is given. And so that background noise cancels out. The other thing we do is in our clinical study, we don't study patients who really, and most of these patients aren't really eligible for chemotherapy anyway, but patients with conditions where there's high levels of necrosis and apoptosis from non-cancer sources, such as very severe inflammation and patients with infections. But the third thing is that we're really excited about is that we can filter out cfDNA from those sources by looking at that fragment length. As it turns out, cancer really sheds DNA that is fragmented into these much shorter fragments. And so by looking at those short fragments, we're able to really filter out non-cancer sources of cfDNA. And in our studies, we actually spiked, if you will, using white blood cells, the cfDNA levels to the point where the amount of cfDNA went up fivefold. And when we filter out those long fragments, the cfDNA level is much lower, and exactly what you would expect from a patient who is not progressing. So we really have good evidence to support that these three methods really do get down to cancer-specific cfDNA.
Well, and we have some great questions from our audience. Again, I'm hopeful that those of you asking these questions will join us in the networking, because these are getting pretty technical, but I'm going to throw them out there anyway. I know. So, do what you can. And if you want to say, we're going to delay until networking, you can do that. Interesting concept. Definitely something which is needed in the field. The question here is, there have been some studies that show that the correlation of cfDNA with radiological progression is still an imperfect science. They put a study in here highlighting that evidence. Can you speak to that a little bit?
Sure. So, I mean, our studies certainly show that we can do this with a high level of specificity. And specificity is really the most important metric for this field. So, the idea here is that physicians have to have confidence that when we identify with a Librix a patient who is progressing, that in fact that patient is progressing and their therapy is not effective. And that's the most important metric. And so what we've done here is in all of our studies, and going forward, we will optimize the assay for specificity. And we have been able to identify so far 100% specificity. And the methods that we use are very different from other cfDNA companies, which is why we've been able to achieve a 100% specificity. If you want, additional information on it, I really do encourage people to come to our networking session, and our medical chief medical officer and chief scientific officer can answer the question probably in more detail than I can.
No, I thought that was great, and your data are certainly very impressive. So another question, and I'm just going to read a little bit, but the problem with stage 4 cancers is that most of the time the treatment options may be limited, and that switching therapy too early might actually disable patients to receive benefits from therapies which may take longer to shrink tumors. Can you address that concern?
Sure. So the physicians we've spoken with and the evidence that we've seen, especially from our MD Anderson study, where we took serial tests and we were able to see how a patient was progressing and how we were able to identify patients whose cfDNA levels were going up or down. And in no case there did we see a patient who was doing progressing, and then in further on down the line, their disease started to they started to respond to therapy. So if a patient's going to respond to therapy, it's going to happen from first therapy on. There are certainly some studies initially with immunotherapies where it appeared as though patients did not respond initially and then later on started to respond. But further studies have suggested that these are really patients who are exhibiting what is known as pseudo-progression. And in fact, they weren't really progressing. It was simply a facsimile of imaging that was picking up inflammation around the tumor. And again, I think Dr. Knopf can address that question better than I certainly can.
Awesome. Well, I'm going to encourage everyone in our audience to come to networking. And I certainly will be at your table to hear more answers to these questions. I would love to hear from you. It seems like this assay would also be able to answer questions like, could the patient take a drug holiday or a treatment holiday? How long do they need to be on the treatment? And again, I realize these are probably things that would need to be studied more, but can you address that a little bit?
No, we certainly agree that really any sort of de-escalation of treatment, our assay will be able to help address those questions. Like, for example, take a drug holiday, observe them with this test to see if they're progressing and progressing in an aggressive way, and get them back on treatment. Also, potentially try to take out one of the therapies in their in the bucket of therapies that they're on, and take one out to see what's going on. So there is opportunity to experiment with treatment within obviously reason, and observe what's going on in a real-time manner. So I quite agree with you that there are some very interesting opportunities for therapy de-escalation.
All right, so one last question for you before we move on to our next presenter. Who are your competitors for this type of monitoring? And what are your advantages over your competitors?
Sure. So probably our biggest competitors, potential competitors, are Guardant Health and Natera, two very large cfDNA, ctDNA competitors. And I mean, right away, the biggest advantage is data. To my knowledge, one is they're keeping the data that they've generated very close to the vest, and I have no idea. And I've asked physicians who are close to their company, what are the performance characteristics of their test in the therapy monitoring area, and no one seems to know. So the other thing is the Natera, which does have a publication, their treatment was, or their second blood draw was taken after the second cycle of treatment. And so they really have no data supporting the use of their test in a in a real-time basis, that is after a single cycle of therapy. And then other advantages, much less expensive. Those tests cost in the order of $3,000-$4,000. Their cost of goods is above our intended list price. And then finally, because of the nature of their testing, where they're looking at ctDNA, they have these very, very wide band, if you will, assays that look at many different really dozens of different mutations. And invariably, they have patients who they'll run the test on who they can't yield a result because the particular mutation for that patient is not in their panel. And in fact, the Natera data, they were only able to really yield a result on 75% of patients whom they tested. So
wow,
Yeah. So
Yeah.
yeah, and we were able to generate a result on every patient so far that we've been able to that we've analyzed in our studies. So those are
Fantastic. Those
Thank
are all great things. Again, thank you for being here tonight, Bill. We look forward to meeting you in the networking session. And big thanks to Mo Nori, who is the founder and CEO of ScriptChain Health.
And I am the founder and CEO of ScriptChain Health, a digital health startup that uses AI for cardiovascular disease and readmission prevention. So what's the problem here? The problem here is that there is a very large unfulfilled need to aid physician treating and preventing chronic heart disease that leads to readmission. Readmission costs the United States healthcare system over $71 million a day in penalties and losses. Physicians have reported to state that 80% of them have been burnt out and overextended, and that there's over $101 billion going to annual waste due to overtreatment in the United States healthcare system. So what are some possible solutions to it, right? So our platform offers personalized care as a AI-based clinical decision support platform. We do have a drug and dosage recommendation model associated with it that equates to bettering patient outcomes. All, of course, are 100% automated. That streamlines the physician workflow, and that's integrated directly into it, which equates to lowering overhead costs, increasing bottom line, and reducing over and under treatment. Here is a couple of screenshots that overlook what our UI and UX look like. We made sure that everything is very simple and easy to see. As you can see here, our bar graph to the right is formatted in a percentage format, and to the left is the patient information. Now, let's get into the technical specs of things, right? We do use deep learning. On the left-hand side, the chart outlines some of the leading deep learning models out there that compares using different metrics, such as the DDI rate, which is the drug interaction rate, the Jacquard score, PRAUC, and the F1 score. We found out that PRAUC is the best metric for our specific domain, which leads me to the right graph here. You see here, there is a linear graph here that shows what the outcomes are of these leading artificial intelligence based models. On the far right is ScriptChain Health, and you can see here that we are significantly better than the leading models. I just want to make a little quick side note is that we still haven't integrated the NLP and clinical notes aspect of it, which is why we're actually raising capital right now, $1.5 million for our seed round. And a portion of that will go into getting access to clinical notes to better our model's overall scores. We'll be discussing that in the next slide right here. So let's dive in a little deeper into the science and medicine slide. The DDR model, which is the drug and dosage recommendation model, has structural correlations, temporal dependencies among clinical data, as well as significant medical factors that have been integrated into our model that show that have a significant role into increasing the odds of cardiovascular disease and readmission, such as age, sex, gender, biochemistry, blood glucose levels, cholesterol levels, smoking, diabetes, so on and so forth. Now, the readmission model, right? We also have shown that high iron levels and high cholesterol levels play a significant role into increasing the odds of readmission. And just like I said before, clinical notes is a very important factor into increasing our outcomes of our deep learning model. When we do get access to clinical notes, our performance outcomes are supposed to increase by 25% of what we are today. Significant improvement right there. Now, what's our pathway to market? So we are known as a software as a medical device tool class 2. So we do have to go through the FDA and fill out the 510(k) form in order to go to market. So we're looking to go to market by spring of 2023. After successfully launching and completing the pilot program, which is what we need your help with, to see if your hospitals are open into running a pilot program using innovative solutions. And then once we are going to market, that's where we start to bring in some more revenue into the company. A little bit more about myself and the team, of course. I am the founder and CEO. My background is in tech and biotech. A little bit more about my team. We are consisted of artificial intelligence engineers, software developers, product designers, a business analyst, and we are under the advisory of renowned cardiologists that work at the Brigham and Mass General Hospital. So with your help, we're looking into partnering with you to launch and complete a successful pilot program, as well as complete and cap out our seed round, which is $1.5 million that'll be allocated over to R&D, legal expenditures, as well as business and marketing fees. Thank you so much.
Great. Big thanks to Mo Nori and ScriptChain Health for being with us here tonight. Great presentation, Mo. I am going to start out with a couple of questions, but again, I invite our audience to put any questions you have for Mo and ScriptChain Health in the Q&A, which is just to the right of the chat. So after hearing about your company, I would put this in the category of precision medicine leveraging AI and deep learning, and you're trying to prevent costly hospital readmissions related to heart disease. Is that correct?
Correct, as well as overtreatment and undertreatment.
Fantastic. Tell us a little bit more about what is currently feeding the AI model.
Gotcha. So we have a couple of data sets licensed from Beth Israel that go into our algorithm. So currently we use data points within the EHR system using the FHIR API that stems from vital signs, medication, past diagnosis summaries, age, gender, sodium levels, so on and so forth.
Great. And what I heard as you were talking about your algorithm is currently you're able to make recommendations on drug classes that should be used for a particular patient, as well as dosages, and then also doing some predicting of risk of readmission. Tell us what you're hoping this model will be able to do in the future.
Of course. So we have a couple of things right now. So in the near future, we're looking into making sure that our DDR model and our recommendations are as robust as possible. So being able to recommend the top 2 or top 3 best drugs for that specific illness for CHF, hypertension, and AFib, as well as like the dosage that goes along with it, as well as the ACC guidelines that will be hard coded into it. We also have risk factors in a percentage format for cardiovascular disease, like the 3 that I just mentioned, as well as readmission. That's within like the near future. Long about let's say 4 to 5 years from now, we're looking into dominating the entire industry within CBD, and then moving on to other domains such as oncology.
Very exciting. So a couple of questions, and I'm just going to combine with some from our audience here. You got a great presentation, really exciting. To talk a little bit more about integration into the clinical workflow, and what do you think the biggest hurdle for adoption of this product will be?
Of course. So our integration right now, we have integrations with the big 4. So that's Epic, Cerner, Athena Health, and Allscripts. But we have 2 ADP partners with the two out of the 4 EHR systems. We will be located in the app orchard of the EHR system, so you don't have to go through a different browser to be able to open our platform. You can actually open it directly within the EHR system, which fits within within the physician's workflow, and making sure that we try to build synergies between us and the EHR systems. So we're not looking into replacing EHR systems at all. We're looking to work with them. As far as like adaptability goes, I mean, we sent out a survey nationwide with over 200 cardiologists that responded to our surveys, and their biggest pain point was to making sure that our tool is as easy to use as possible. So
Mhm.
usability is a number one factor for us. If we make sure that our platform is very easy and robust to use, building it as less clicky as possible too, we believe that we can build the trust with these physicians, as well as build our platform with the gold standard of the ACC guidelines.
That is super exciting. I agree. Somebody just gave us a clapping hands emoji, which is awesome. One thing that I was thinking about, because the proof is in the pudding in the end, right? So are you planning, especially as you kind of expand the algorithm, saving money by preventing those costly hospital readmissions is going to be what will sell your product or make your product sell itself. So are you planning any additional clinical studies moving forward?
Definitely. We're looking into launching a couple of pilot programs, and with the audience's help, I mean, if we can get a physician or two who would be interested into, piloting and being our champion at their hospital or at their clinic, we're 150% open to it.
Awesome. That's what I love. Okay, so a couple of questions that are now getting upvoted. So that means that more than one person in our audience is happy to hear. In the future, do you envision integrating data from smart wearables? Because really, that might be the future of medicine, huh?
You do have a really good point there. That may be possible. That may be possible as part of our roadmap.
That would be awesome. I know, even though I'm a pediatrician, I know those regular weights on heart failure patients are really important. So I could see some of that happening. That'd be very cool. We also have one additional question. I know you mentioned that you have a Fire API. Can you talk more about that? And I'm guessing that's going to help you scale dramatically in the future. So talk a little bit more about that as well.
Of course. So right now, the EHR systems have been pushed by the government to making sure that they're able to build interoperability between different EHR systems. Where they built the Fire API, right? It's HL7 as well as Fire, but we're going with Fire just because that's going to be the future standard of integration with these EHR systems. With building like the different data points within the API, we're able to integrate it very quickly. And we are a cloud native application too, so we're able to scale fairly quickly. The data points are very similar from EHR system to other EHR systems. So making sure that the implementation phase of our platform within the EHR system is as seamless as possible, as well as like shorten like the bandwidth of implementation, which is very important to us.
Very cool. Well, Mo, I would have to say, huge problem that you're working to solve here. So very exciting. I'm looking forward to hearing more about what ScriptChain Health is doing in the future. And thank you again for being here with us this evening.